the anatomy of the kidney structure. So now let's look and see how it works, how it functions. So these are the basic principles of renal function. Now we know that the kidney filters the blood, but it's not filtering the blood like the spleen. The spleen is taking out old and or damaged red cells. The kidney is actually filtering the blood. It's actually filtering the plasma of the blood. All right? So in that filtrate is just not stuff that we need to get rid of. Everything's in there. All the nutrients, the water, as well as metabolic waste and other stuff is in there. All right? So the kidney filters that, so it conserves what's useful to us. It takes back all the nutrients and most of the water out of that filtrate. And it removes the metabolic waste in the process. Now, many of the waste products in the body are dissolved in solution. Okay, so the kidney has to be presented with stuff that's in solution. It can't act on dry material. So it has to be in solution. And therefore dissolved in tubular fluid or urine. All right, here are the ways that the kidney works. These are the three main mechanisms by which the kidney works. Filtration. Okay, all the filtration happens in the renal corpuscles. Okay, then reabsorption. Reabsorption happens in the tubular system and the collecting duct. Okay, so reabsorption is taking back out of the filtrate what the renal corpuscles have filtered out of the tubular fluid. Secretion is basically a backup to reabsorption, because some of the things we get rid of are secreted, not necessarily filtered in the renal corpuscles. Okay, so reabsorption works like this. You got stuff in solution in tubular fluid. It's going to pass into a tubular cell and into the fluid around the cell, interstitial fluid, and into blood. That's reabsorption. Secretion is the opposite of that. You've got something in the blood that the body needs to get rid of. It passes out of blood, into interstitial fluid, into tubular cell, into tubular fluid, and out. Okay, so secretion doesn't happen just in the kidney only. You know, it can happen in other organs too. So we're studying, you know, in lab now, we're on endocrine system. These endocrine glands secrete their hormones. Okay? It's a process of secreting, moving out of something into something else. All right, so let's get some terms straight. Tubular fluid and peritubular fluid. So this filtrate of plasma that happens in the glomeruli, in the renal corpuscles, that filtrate passing into the tubular system, we're going to refer to that as tubular fluid. It's not urine yet until it gets well into the tubular system, down, say, into the end of the distal convoluted tubule and in the collecting duct. At that point, we can call it urine, but it's still not going to necessarily have the same concentration and composition as what shows up in the bladder, because on its way down the collecting duct, more water can be taken out, and various things can be secreted into it, various other things can be reabsorbed from it. So we can call it urine, but it's not going to have its final composition and concentration until it drips out of the end of the collecting duct via the renal papilla into the calyceal system. Then for sure it'll be what's going to show up in the bladder, same as what's going to show up in the bladder. Peritubular fluid is just interstitial fluid. Peri means around. So it's the fluid that bathes the outside of the tubules. So it's peritubular fluid. It's the same thing as interstitial fluid. Now, let's just get an idea of how efficient the kidneys are at their job. Our kidneys make up about roughly 1% of our total body weight, excuse me, but they receive about 1/4 of our cardiac output. Now, for purposes of our example, let's assume that we've got a total blood volume in a person of 5,000 milliliters. So that would be maybe a normal adult male, 5,000. And we'll say that this plasma volume of that total volume is about 55%. So in this case, our plasma volume would be about 2,750 milliliters. All right? Now, the kidneys filter at the rate of about 120 to 125 milliliters per minute. All right? So if you take that figure, 125 milliliters, and multiply it by the number of minutes in 24 hours, you'll get some idea in our example of how much fluid, how much filtrate the kidneys are producing in 24 hours. You see that? It's 180,000 milliliters, or 180 liters, in 24 hours. That's a lot of fluid, isn't it?
Yeah.
All right? So, in our example, this is equivalent to filtering the entire plasma volume more than 60 times in 24 hours. Actually, it's more than 65 times in 24 hours in our little example right here. That's quite a job, isn't it?
Yeah.
All right, so let's take this in sequence now. The first thing that's going to happen is filtration. And that's going to happen in the renal corpuscle. So up here in our general diagram here, filtration is happening up here. So in our little key, A is filtration. So here is an interlobular artery. Here's an afferent arteriole, glomerulus, efferent arteriole. Here is the capsule here, and here's a subcapsular space. All right, so we're filtering this blood. Okay, so the filtrate is going to be plasma-like, isn't it? In fact, it's going to be plasma except for large molecules like proteins. The large plasma proteins. Okay. So filtration occurs only at the renal corpuscle. And specifically, it's the glomerulus that's doing the filtering. All right. Now, hemodynamics of this process. The glomerular capillary is fenestrated. All right. Remember the fenestrated capillaries? What does this tell you right away?
Allow for more to pass through.
Yes. So filtration is going to be easier, isn't it? All right. Here's another thing. The pressure in the afferent arteriole going in is higher than in the standard peripheral capillaries of the body otherwise. If you remember when we talked about blood vessels and circulation, we said that the arteriolar pressure going into a capillary is around 35 millimeters of mercury. The pressure in afferent arterioles is around 50 to 55 millimeters of mercury. So we got a higher pressure going in, and remember we said that this capillary is not only fed by an arteriole, it's drained by an arteriole.
Why is it a higher pressure?
For purposes of better filtration.
Okay.
Yeah. And because of this.
Because it also has to go.
If we got an arteriole draining the capillary, that arteriole has smooth muscle in it, doesn't it?
It's got more
And
to.
so, as you narrow the lumen of that, you increase the filtration pressure, don't you? Yeah. There's another reason for that down the line that I'll mention, but
Okay.
anyway. Now, this glomerular pressure is a hydrostatic pressure. Hydrostatic pressure is the pressure of a fluid within a closed system. All right. Now, this filtrate that's produced in the subcapsular space, it creates a back pressure of its own because there's fluid down in this proximal tubule. So this proximal tubule is not empty, you see. And so there's some back pressure created as that filtrate accumulates within that glomerular capsule, like this. So here's our afferent arteriole going in. Here's our glomerulus. This here would be the subcapsular space, and this is where the filtrate is going to appear, and then it's going to head down the proximal convoluted tubule. Now, I'm changing these numbers a little bit in your notes. I'm just rounding them off some. So I'm going to, for purposes of our calculation, I'm going to say that the glomerular hydrostatic pressure is 50 millimeters of mercury, and the blood colloid osmotic pressure is 25 millimeters of mercury, because when we were talking about peripheral blood vessels, we labeled it as 25. So I'm going to call it 25, and the capsular hydrostatic pressure will remain at 15. Okay. I couldn't change these numbers on this particular figure right here. So, what is our net filtration pressure? And that's what this screen is showing you right here. So we got our pressure going in. That's the glomerular hydrostatic pressure. We've got some pressure of the fluid that's produced as the filtrate, capsular hydrostatic pressure, which is going to be opposing this. So this fluid that is accumulating in here, if you look at your green arrow, it's pushing back against the glomerular hydrostatic pressure that's filtering the blood. All right, so our net hydrostatic pressure is going to be this minus this.
So 10.
All right, now existing within the bloodstream is this pressure, the blood colloid osmotic pressure. This is caused mainly by the plasma proteins. This number is going to be 25. All right, so our net filtration pressure is simply going to be the difference between this and this. See? So here's the way we would calculate it. If we assume our glomerular hydrostatic pressure is this, our capsular hydrostatic pressure is this, and our blood colloid pressure, osmotic pressure is this. So here's the way it works. 50 minus 15 is 35, isn't it?
Yeah.
35 minus 25 is 10. That's our net filtration pressure. Now, notice, folks, this is a low pressure. 10 millimeters of mercury, that's hardly any pressure at all. But it works. Of course, now, if we have things that obstruct the flow of urine, or things that create pressure in a kidney like infection or inflammation or trauma and bleeding, we can easily resist this pressure, can't we? And it can affect renal function. If you have a stone that's blocking a ureter, and urine is backing up in that thing, backs up in the ureter, backs up in the calyceal system, puts pressure on the parenchyma of the kidney, that's going to limit the ability of the kidney to filter, isn't it?
Yeah.
All right, so what we're getting around to is the glomerular filtration rate. And as I mentioned earlier, it averages around 120 to 125 milliliters per minute. And remember, in that filtrate is everything except the large plasma proteins. We got to take back what we need and pass the rest on down. All right, as this is what I was telling you, this filtration rate can be adversely affected by anything that overpowers the net filtration pressure in the kidney. Here are some examples that we mentioned. So, for example, you take this one right here. Someone is experiencing traumatic injury, has lost a lot of blood, they've got a low circulating blood volume and a low systemic pressure. If you've got a low systemic pressure, isn't that going to affect the blood pressure going into the kidney through the renal artery?
Sure.
And isn't that going to affect
Function.
the branches of the renal artery and ultimately affect the pressure in the afferent arteriole? Yeah. And so, if you've got an uncorrected, profound hypovolemia and hypotension, this could result in renal shutdown. Could. In fact, when a person like this comes in the emergency room, one of the things that's done is to put a catheter in the bladder to monitor urine output. And these folks who've lost a lot of blood and are hypotensive in, circulatory shock, they're not making much urine at all, if any. So this is why you've got to supplement. You've got to give them IV fluids, sometimes plasma expanders, sometimes blood, to get that pressure back up.
Now, is that an extreme case of hypotension? What if someone just on average has low blood pressure, that their blood pressure just runs low normally?
No.
That
If their pressure is low normally, they're still going to.
So is this more
have enough
extreme
pressure.
case, or just a shock to the body that's going to? Okay.
Yeah. Because somebody that has a low blood pressure, what's a low blood pressure?
My daughter, it's something like 70. It's really low. It's
Yeah.
very, very low.
Okay, but the mean pressure in the aorta, by the time that blood gets out of the heart down to there,
Okay.
is going to be sufficient to
Do it.
supply the kidney.
Okay.
Now, the kidney has mechanisms by which it can regulate its glomerular filtration rate when it's adversely affected. Okay, so it has ways to do this. So it can adjust its own resistance to blood flow and therefore maintain a nearly constant glomerular filtration rate despite fluctuations in blood pressure. So there's another thing that would help your daughter, you said? It runs at kind of a low pressure. So there are two ways the kidney does this. Something called intrinsic control and something called extrinsic control. But let's look at intrinsic control first. Intrinsic control is in operation when the volume of extracellular fluid is normal, when blood pressure is within normal range, and when the sympathetic division of the autonomic nervous system is at rest. Okay. And extrinsic control, this is not the case, but this is your day-to-day operation right here. This is normal. This is what happens with you normal, all of us, day to day. Now there are two aspects of this. One is called myogenic mechanism. The other is tubuloglomerular mechanism. But by either one of these or both, the bottom line is that the kidney regulates its filtering rate to optimize tubular reabsorption of solutes and water. Okay, so the kidney not only has to filter it, it has to get it back. But it can't get it back unless it filters it first. So on this chart right here, intrinsic control is this much of the chart right here. This much. So the first mechanism is myogenic. When blood pressure drops in afferent arterioles, the vessels leading into the kidney dilate. And when you dilate a vessel, you increase the flow, don't you? You increase the flow, you increase the pressure. And so this will up the glomerular filtration rate. Now, another somewhat trickier way is this way, tubuloglomerular mechanism. Now, let's run back to our juxtaglomerular apparatus. Remember the distal convoluted tubule? It swings back up close to the renal corpuscle. When it does, there are some specialized cells in the distal convoluted tubule right here called the macula densa cells. Those cells have the ability to monitor the fluid flow rate coming past them. They also have the ability to measure the solute concentration of that fluid that is flowing past them. All right, so when the fluid flow rate drops in the fluid that's flowing past here, that means that the glomerular filtration rate has dropped because we're producing less filter, therefore less tubular fluid to flow through this tube. All right, when you slow down the flow of tubular fluid through the convoluted tubular system, that gives more time for reabsorption of solutes, sodium and other things. And so these macula densa cells notice when there's a drop in solute concentration, drop in flow rate, a drop in solute concentration of that fluid. That's what's happening right here at the juxtaglomerular apparatus. And so that's what we were talking about right here. I should have put another picture in there instead of having to flip back and forth. So you see right here, that's what it's saying. A drop in the flow rate, a drop in the solute concentration, that's mainly sodium chloride, causes vasodilation of afferent arterioles. You dilate the vessels, you increase the flow rate, you increase the pressure going in, you'll increase the glomerular filtration rate. Now, oh, there it was.
Oh, no.
There it was. I did put it in there. All right. Oh, well. Now, the other method is extrinsic control of glomerular filtration. This applies when there's been some major insult to the hemodynamics in the body. So you've got a drop in circulating blood volume, a drop in pressure, or both. Now we have to shift to another gear. And we've got to call on some external means to increase the glomerular filtration rate. The kidney is limited in what it can do. So the extrinsic control is both neural and hormonal. The neural part is sympathetically stimulated. The hormonal part involves this system right here, and later this. All right? So, notice that this is operational when systemic blood pressure is markedly compromised. So we got a drop in pressure to somewhere less than 80 millimeters mercury systolic. And when that happens, we get a sympathetic discharge. Okay, so we know what happens when sympathetic discharge occurs. On the medullary centers, it increases heart rate, force of contraction, increases peripheral resistance to bring the pressure back up. We also now bring into play the renin-angiotensin II mechanism. And the reason that happens is when there is a drop in pressure going into the afferent arteriole, these cells right here, the granular cells, release the enzyme renin. Remember we talked about this? Renin triggers the release of angiotensinogen from the liver. Angiotensinogen is converted to angiotensin I. That's not the active form. Angiotensin I has to be converted to II. That happens in mainly the pulmonary capillaries, via an enzyme called angiotensin converting enzyme. Angiotensin II is the active form. Angiotensin II itself is an extremely potent vasoconstrictor. It's about 8 times as powerful as norepinephrine. But angiotensin II also causes some other things to happen. It causes the adrenal cortex to release aldosterone, a hormone that promotes sodium reabsorption by the tubular system. As sodium is reabsorbed, water follows it. So water is added back to the circulating blood volume. It also triggers the posterior lobe of the pituitary gland to release antidiuretic hormone. Antidiuretic hormone acts on the collecting duct to promote water reabsorption. So the body is trying to recover fluid to increase circulating blood volume to raise blood pressure. That's what's happening with this extrinsic control here. And that's what this is telling you right here. This is all in your notes. It also increases thirst. If you're ever working in the emergency room and you happen to be there when somebody comes in profound circulatory shock and they're still lucid, they will often say, "Can I have a drink of water?" Thirsty. And that's because of this right here. Now, so this part of our chart here has to do with extrinsic control. So, angiotensin II, a potent vasoconstrictor itself, acts on the adrenal cortex. Now, this particular diagram, I must tell you, doesn't show the effect of antidiuretic hormone. I'm going to show you about this later on, but I don't know why they didn't include this on here, but they should have. This is sympathetic discharge here. Sympathetic discharge can also trigger the release of renin from those granular cells. Now, this is the other hormone that's involved in extrinsic control. Let's say that somebody comes in the emergency room and hemodynamic compromise, low circulating volume, blood loss, low pressure, and we have these mechanisms in play by the body itself to try to restore homeostasis, but it usually takes more than that. So medical personnel have given some IV fluids, maybe plasma expanders, maybe blood, to get the circulating volume up and the pressure up while they're investigating what the cause of this is and what needs to be done to fix things. All right, but let's say that resuscitation has been successful, and let's say that there's been an overcorrection. Too much IV fluid, too much plasma expander, pressure's up. So as you increase circulating blood volume, you increase the volume of blood returning to the heart, don't you? You increase venous return, don't you? As you increase the volume of blood returning to the heart, it stretches the right atrium. And stretching of the right atrium releases this hormone, ANP, atrial natriuretic peptide. This hormone, the easy way to remember what this hormone does is it does the opposite of what angiotensin II does. It cuts off angiotensin II. So it cuts off vasoconstriction, it cuts off aldosterone, it cuts off ADH. Right. And now, if we got an overcorrected circulating volume, here's what this hormone will do. It dilates the afferent arteriole and constricts the efferent arteriole. Now, what does that do to the glomerular filtration rate? What does it do to the hydrostatic pressure, the glomerular hydrostatic pressure? It's going to increase it, isn't it?
Yeah.
So it's going to increase the glomerular filtration rate to get rid of the excess water. Also, if too much sodium has been given in the process of resuscitation, this hormone will promote sodium loss via the kidney. In other words, to get back to homeostasis. Normal circulating blood volume, normal sodium content. All right, so that's filtration. Now we're down to tubular reabsorption and secretion. Okay, so out of this filtrate we need to recover all the nutrients and most of the water. Now, back to filtration just a minute. We said that the plasma proteins remain in the circulating bloodstream. The large plasma proteins can't get through this filter, right?
Right.
Some of the smaller plasma proteins sometimes can get through. But if they do, the proximal tubule promptly reabsorbs it. So it's not normal for protein to show up in the urine. So if protein is showing up in the urine on a chronic basis, something's wrong in the kidney somewhere. So we're down here now with B, tubular reabsorption. We've got to get back to things that the body needs. So some general principles. Most of the water and solutes in the filtrate are reabsorbed, as we mentioned. These absorbed substances pass through several membranes here that I'm going to show you. So reabsorption is in this sequence. Secretion is in the opposite sequence. So, right here. This is absorption. This is tubular fluid. This is tubular cell. This is peritubular fluid or interstitial fluid. This is blood. So things that are reabsorbed pass through the apical part of the cell. The apical part is the part facing the tubule, lumen. Okay, so they pass through the cell, out the basilar part, in of the cell, into peritubular fluid, into blood. Things that are secreted pass out of blood into peritubular fluid, into the basilar part of the cell, through the cell, and out the apical part, into the tubular lumen. Now, the processes involved in reabsorption. Diffusion. Diffusion is the moving of things from an area of higher concentration to lower. Water moves the same way. Water actually moves by diffusion, but we call it osmosis. Right? But many things, in fact most things, pass through cell membrane through protein carriers or channels. Here's some examples. This is simple diffusion of lipid-soluble substances through the cell membrane. These are all examples of protein carriers or channels through which things move into and out of cells. As far as this one goes, the carrier-mediated transport, here are some subcategories of that. Facilitated diffusion is things moving down a concentration gradient via a carrier protein. Doesn't require energy. Some things do require energy, like the movement of sodium and potassium. Sodium is mainly an extracellular ion. Potassium is mainly an intracellular ion. So the cell pumps sodium out of itself into extracellular fluid in exchange for potassium, which moves out of extracellular fluid into the cell. This is called an ion exchange pump, and it does require energy in the form of ATP. Co-transport just means that two substances are moving together in the same direction. For example, glucose moving into the cell commonly moves with sodium. That's just co-transport. Counter-transport is when you have two substances moving in opposite directions at the same time across the cell membrane. A typical example is bicarbonate and chloride. They always move in opposite directions across the cell membrane. All right, so let's head down the convoluted tubular system and see what happens. Proximal convoluted tubule. Notice, most of the reabsorption occurs here. And just what do we reabsorb here? Notice, most of the water, 60 to 70%. Now, as we go through the tubular system and the collecting duct, we're going to keep a running total of how much water is reabsorbed where. So for purposes of our calculation, we're going to say that 60% of the water is reabsorbed in the proximal convoluted tubule. All right. What else do we reabsorb? Organic molecules and various ions. So you see that the proximal convoluted tubule is basically reabsorbing virtually all of the nutrients and by far most of the water. All right. Now, we need to talk about this because this is an important principle here. It's called the transport maximum or tubular maximum. There is a maximum amount of any substance that the tubular system can reabsorb. In other words, for every substance in the body, there is a maximum that can be reabsorbed in the tubules. For everything except sodium. There's no maximum for sodium. So, for example, glucose. There is a tubular maximum for glucose, just to give you an example. A normal person can absorb all the glucose that's presented in the tubule. A person who, say, is diabetic and uncontrolled and has high blood sugar is going to be passing a lot of glucose in that filtrate and thence in the tubular fluid, isn't it? That person can only absorb up to a certain point the glucose that's presented to those cells. Anything over the maximum concentration that can be reabsorbed is going to pass on down the tube and will show up in the bladder and the urine. Okay, so this is what we mean by the tubular or transport maximum. So, in our course down the system here, this is where we are right now. So, this is here. So this is what's happening in the proximal convoluted tubule. It's a busy place, isn't it? So this is an example of the kinds of things that happen in the proximal convoluted tubule. Here's tubular fluid, tubular cell, peritubular fluid, blood. So notice that, here's an example of co-transport right here. Sodium and glucose are moving in the same direction. We're absorbing amino acids, ions, vitamins, some urea, although we'll get rid of some urea down the way. Fat-soluble substances, water, and so forth, various ions. Now, notice here that these things require a protein carrier. Here's an example of an ion exchange pump right here. Notice our little key here says that we require energy for this. This is active transport. It requires ATP. This over here is secondary active transport. See the dotted line?
It doesn't require ATP.
This means that some energy is required, but it's a spin-off of this energy right here. So the movement of glucose into cells does take some energy. Now, some substances are not reabsorbed or are incompletely reabsorbed. Why is that? Well, it could be because they lack carriers, or because they're not lipid soluble, or because they're too large to pass through the membrane pores. Here are several important substances that are not reabsorbed or are incompletely reabsorbed. So, urea. What is urea?
Just waste. Just waste.
Ammonia and CO2.
Right.
Waste.
It is the detoxification product of ammonia. The liver combines ammonia with CO2 and produces urea, which is non-toxic. All right? Now, we get rid of most of that via the kidneys, but not all of it. We retain some in the bloodstream. So if you've ever had any kind of basic chemistry done as a blood test, one of the things they do is a BUN. Stands for blood, urea, nitrogen. It's normal to have low levels in the blood. All right, creatinine is practically all not absorbed. A little bit is. Most of it is not reabsorbed. A little bit is. So it's normal to have a low level of creatinine in the blood. Now what if you have elevated BUN, blood urea nitrogen, and or elevated creatinine? What's that a sign of? Yeah, this is a sign of impairment of renal function. All right? Uric acid is another thing that's incompletely reabsorbed. All right, that brings us to the loop of Henle. What's going to happen here? We're going to reabsorb water, sodium, and chloride, and a little potassium. How much water are we going to reabsorb in the loop?
25%.
About 25%. So by the time we get out of the loop, we have reabsorbed 85% of the water, right? Now, there's some distinctive features of the loop's function, and that is this, that the descending limb of the loop is permeable to water, but not to solutes. And the ascending limb is permeable to solutes, but not to water. One of the main functions of the loop of Henle is to concentrate the urine. I'm going to show you how this works in a little bit. So the only thing that's happening in the loop now is reabsorption of water and certain electrolytes, sodium, chloride, and potassium. So this is where we are now in the loop. That's what's happening here. Now, here's an example of what happens as we go through the loop. So here is our renal corpuscle, here's our proximal tubule, and now we're in the loop here. Now, this number, 300 here, represents the solute concentration of blood, or the osmolality of blood. Actually, it's 295 milliosmoles per liter, but we just rounded off to 300. All right, so as we start down the proximal tubule, the osmolality or solute concentration of tubular fluid basically matches blood, doesn't it? But as we go down now, we are reabsorbing water out of the proximal, out of the descending limb. So what's that doing to the solute concentration as we go down?
It's increasing it.
It's increasing. So when we get down to the bottom here, it's 4 times what we started with. All right. Now as we come back up, you notice the numbers are decreasing, aren't they? Why is that?
Because the water's moving back in.
Because now the ascending limb is permeable to solutes, but not to water.
Right.
So we're getting rid of solutes.
Water's gonna get
And
rid of them.
even to the point that as we come out of the loop, we've got a lower solute concentration than we started with over here. Unless something happens in the collecting duct, which I will show you a little bit later. Now, this over here represents the vasorectum. The vasa recta is the capillary that's associated with these long loops of Henle, of these juxtamedullary nephrons, correct? All right. What this vasa recta is going to do, and this is the vasa recta right here. Let's go back, show you. What this vasa recta is going to do, it's going to maintain the solute concentration of blood stable. All right. Now you got to think in two different avenues here, because what's happening here in the blood vessel is different from what's happening here. All right. Unless you are thinking of that, this is going to confuse you. But now, as we go down here, you'll notice that the numbers pretty much are matching this, aren't they? All right. So as we go down, we have got both water and solute movement happening. All right. But the solute concentration, the movement of solute, is dominant. And that's why the number is increasing. All right. Osmosis is hindering this process here for water. But as we turn the corner and come back up, we still have solute and water movement, both happening. But now, osmosis is dominant. And we're bringing in more water than we are getting rid of solute. And so we are ending up at the same number we started with. So this maintains the solute concentration of blood stable. Can you imagine what would be going on in the body if our solute concentration was going through these extremes constantly like this in blood? It would be a mess. All right, so that's what the vasa recta does. It maintains a stable osmolality or solute concentration of the blood. And that's what we were just saying. All right, now we're down to the distal convoluted tubule. We're coming out of the loop, we're in the distal convoluted tubule. What happens here? Well, we can reabsorb some water here. But if we do, it's going to be under the influence of aldosterone, that hormone of the adrenal cortex we talked about. Aldosterone promotes sodium reabsorption. And sodium reabsorption in the distal tubule will cause water to come with it. Remember the axiom, water follows salt. So we could reabsorb as much as 5% of the water in the distal tubule if aldosterone is operational. Now, some other things happen in the distal tubule. It's capable of reabsorbing and secreting hydrogen and bicarbonate. It's also the primary site of calcium reabsorption. So this is where we were just now, in the distal tubule. And that brings us to the end of the nephron. So what's dripping into here, we can call it urine, okay, but there's some other things that are going to happen down here in this collecting duct. Before we get to that, let's talk about secretion. Secretion is the opposite of reabsorption, remember? All right. Now, this is a look at secretion in the different parts of the nephron. So, the proximal tubule can secrete these things. Now you look at this and you say, well, I saw that the proximal tubule reabsorbs urea. It does. But it also secretes most of it back into the tubular fluid. Proximal tubule absorbs a little bit of creatinine, a little bit of uric acid. It's capable of taking on hydrogen ions if we're deficient in it. I mean, of getting rid of hydrogen ions if we're deficient in it. Also potassium, ammonium, organic acid, drugs. The loop, no secretory function at all. Distal tubule, it can also work with hydrogen, potassium. It can also get rid of various drugs for us by secretion. All right, that brings us down to the collecting system. In the collecting system, we can both reabsorb and secrete. So, these collecting ducts are receiving tubular fluid from many nephrons. We've seen this. And they're going to take it down all the way through the renal medulla and drip it out the ends of those renal papillae there. Now, what are we going to reabsorb? Well, we can reabsorb some more water. The collecting ducts are capable of absorbing this much more of the remaining water. However, it doesn't happen automatically. It only happens if this hormone is operational. Antidiuretic hormone is a hormone of the posterior pituitary. If it's operational, it will cause the insertion of certain proteins called aquaporins into the membrane of the collecting duct. And that will cause water to be reabsorbed by the collecting duct. So we must have this hormone to cause water to be reabsorbed.
Okay.
What would
Now,
the reason be why that hormone wouldn't be available from the pituitary? Posterior pituitary. Why would that not be secreted at home?
The osmolality of the blood, the solute concentration of the blood. The solute concentration of the blood is constantly monitored by the hypothalamus.
Oh, right.
There's some little osmoreceptors in there. And it measures it within a very narrow range. So let's say that the solute concentration of the blood is drifting a little higher than normal. The hypothalamus stimulates the release of antidiuretic hormone from the posterior pituitary. It acts on the collecting duct to cause water reabsorption. As you reabsorb water, it brings the solute concentration back down. When that's okay, hypothalamus cuts off ADH. All right, the collecting ducts also reabsorb urea. You'll take a look back at page 6 of your notes. You know that little diagram I gave you there? It's not in your textbook. The lower part of that collecting duct is a papillary duct. And I put that in there to show you where urea is reabsorbed. Urea is reabsorbed through that papillary duct. Also, the collecting duct has the ability to reabsorb hydrogen ion or bicarbonate ion, depending on the pH of the blood. So if the pH is on the acidic side, what do we need? We need something to neutralize the excess hydrogens, so the collecting duct will reabsorb bicarbonate. If the pH of the blood is alkaline, we have the opposite. We need hydrogens to neutralize the bicarbonates, and so we reabsorb hydrogen. Now, here is an example of how ADH works. Remember back on our loop, we said that coming out of the loop, we're actually hypoosmolar here. And coming down the collecting duct, that situation would obtain all the way, dripping into the minor cavities. All right. However, if we have antidiuretic hormone operational, we're going to reabsorb water, aren't we, from the collecting duct. And what's that going to do now to the solute concentration of this fluid?
It's going to increase
it. It's going to increase it, isn't it? Now, you see urea being absorbed right down here? You see this 1200? All of that 1200 is not due to sodium chloride. About 750 milliosmoles is due to sodium chloride. The other 450 is due to urea. So the absorption of urea plus the increased concentration of sodium here is going to cause water to move more out of the collecting duct too. So what's happening here in these long loops of Henle, remember we said that the long loops have the ability to concentrate the urine?
Yeah.
So as you pull water out of the collecting duct, you increase the concentration of the urine, don't you? And so this urine that's dripping out here has an osmolality of 1,200 milliosmoles per liter. It's because we're absorbing more water out of the collecting duct. All right, so let's summarize our water reabsorption. Proximal convoluted tubule, 60%. Loops, 25%. Distal convoluted tubule, let's say that aldosterone is operational, 5%. Collecting ducts, as much as 9 to 9.5%. And I didn't put, we'd have to have an antidiuretic hormone in play here. So, the totals for water reabsorption in 24 hours. Back to our example. Let's say that in our example we're filtering 180 liters in 24 hours. All right. So, of that 180 liters or 180,000 milliliters, we recover all but a half. We can recover all but a half to 1% of that total filtrate. So this makes our urine volume for 24 hours to be somewhere in this range, doesn't it?
Yeah.
You see how efficient the kidneys are. All right, secretion. The kidneys, the collecting ducts now not only can reabsorb, but they can secrete. So we said that they can reabsorb hydrogen or bicarbonate depending on the pH of extracellular fluid. They can also secrete hydrogen and bicarbonate depending on the pH of the blood. All right, so either way, got both, all bases covered. It also can secrete potassium and ammonium ion. So this is where we are with this now. This is all the stuff that can happen in the collecting duct over here on the far right. Now, we'll finish up on this. Micturition or the voiding reflex. So, the bladder is a storage organ, temporary storage organ for urine, is it not? All right, here's what happens. Say you empty your bladder. All right. Now the bladder is going to fill again. You've got fluid coming, urine coming through both ureters, and that bladder is filling up. While this is happening, sympathetic input, shown right here, inhibits muscular action in the bladder wall and keeps the internal sphincter tight. All right? So sympathetic input is, at this point, as the bladder is filling, is overriding parasympathetic activity. Bladder contraction and emptying is primarily a parasympathetic function. All right, so as this bladder fills with urine, the stretching of the bladder initiates some afferent or sensory input to the sacral cord via parasympathetic fibers. That results in motor activity, efferent impulses back to the bladder to cause the bladder muscle to contract, and you feel the urge to empty the bladder. Okay, when that muscle contraction starts, the internal urethral sphincter relaxes, but no urine comes out yet. Why?
Because you've got
Because you got another sphincter, and that other sphincter, the external sphincter, is cortically controlled. It's voluntary. All right, spinal nerve, skeletal muscle. Now, while this is going on, while the bladder is stretching and you're feeling the urge to void, the cortex is aware of this. So you know you need to find a time convenient to empty the bladder. So when you do that, you consciously relax the external sphincter. You've overridden the sympathetic input to the bladder. It's all parasympathetic now. And you consciously relax the external sphincter and allow the bladder to empty.
Now, with someone who's had a spinal injury, they no longer have that voluntary release then? I mean, even though they have cognitive, let's say it was a
Right.
spinal injury below the waist where you have all your cognitions, you can't voluntarily just say, okay, I go to the bathroom, let me try. It just doesn't work anymore?
They don't have the connection.
What about a person that's paratonitic and have stents in? Is that a kidney stone? And I was wondering that I didn't know why it's that.
Probably irritation from the stent. Okay. Oh, almost forgot the terms. Here are a few terms you need to be familiar with. Oliguria, O-L-I-G means few or scant. So oliguria is scant urine production. Anuria means no urine production, right? A or an in front of a word means absence of.
Right.
Dysuria, anytime you see dys in front of a word, it means bad or painful. Okay, so dysuria is painful urination. Hematuria, obviously, is blood in the urine. Pyuria, pus in the urine. Pyelonephritis is a term that usually refers to a bacterial infection in the kidney. So pyelonephritis. However, it's not the same thing as glomerulonephritis. Glomerulonephritis is not due to a bacterial infection normally. It's more commonly an autoimmune response that damages the glomerulus. All right, and so it's not the same thing as pyelonephritis. Cystitis is an irritation or infection in the bladder. Anytime you see itis on the end of the word, it means inflammation of. Lithiasis is just a term that refers to stone formation. So if you form stones in the kidney, that's nephrolithiasis. Stones in the ureter, ureterolithiasis. Stones in the bladder, cystolithiasis. Hydronephrosis is a term that refers to dilatation of some parts of the urinary system. It usually is a result of some distal obstruction that causes dilation of the ureters, and that back pressure dilates the calyceal system